Yes, dear students of TYB Pharmacy, today we are going to discuss about the subject that is Medicinal Chemistry 3 theory, and the name of the chapter is Introduction to Drug Design and Combinatorial Chemistry. Now here we are going to discuss today about the drug design concept and what are the applications of this drug design in the pharmaceutical industry. So in the earlier days, purely randomized search was carried out at that time, and the procedures were involved in the discovery of new drugs. So in earlier days, the random technique is used for the procedures involved for the discovery of new drug. But these methods, the experience and the intuition of the medicinal chemist were the important factor to reduce the stochastic nature of these search techniques. And in a view of the same, the increasing number of chemical compounds, due to the population there is increasing number of the chemical compounds and the drug, and in particular the heavier demand which is not made by the new compounds, which we are going to search at that time by random method. So in a view of the same, there is it is too time consuming process, and there is no any guarantee that little success, and the drug which is going to be produced is too expensive in nature. So in a view of the same, there is a chance for we will go for the medicinal chemist will go for the drug design and combinatorial chemistry technique for the search of new drug designs. And in view of the same, the drug design. Now what is basically actually a drug design? So drug design is the integrated technique for developing the protein era of tailoring drug, and which will involve the study of effect of biologically active compound on the basis of molecular interaction in the terms of molecular structure. So in this particular drug design concept, we are going to study the integrated developing discipline technique, which is known as a tailoring drug, and this technique involves the study of effect of biologically active compound on the basis of what is the structure of that drug molecule, and along with that structure of drug molecule, we will also know the physico-chemical properties which are involved, which are associated with that drug molecule. And in a view of the same, the objective of this particular chapter is to understand the importance of the drug design and what are the different techniques which we can use for the drug design concept or the application of the drug design, and to know the importance of SAR belongs to structure activity relationship of this drug molecule. Now what we learn actually in this particular chapter, so the learning objectives are: student will learn about the different techniques of the drug designing. Student will also learn about what is the relation of the drug and its activity with respect to the physico-chemical properties of that drug molecule. Now we will go in depth about what is actually a quantitative structure activity relationship, which is being a part of drug design concept. Now the content of this particular today's chapter is introduction, hydrophobicity of the molecule, hydrophobicity of the substituent. Now both of these points, that is hydrophobicity of the molecule, that is parent molecule itself, in which we will go for the different type of substitutions. So we also know the physico-chemical properties of that substituent as well as parent molecule. Now next is what are the electronic effects offered by that molecule, then steric effect, when how the molecule is going to be behave in the space, what is the stereoelectronic configuration of that molecule, and followed by we are going to discuss a very important equation with respect to the physico-chemical properties of the drug, and the name of that equation is Hansch equation. We will also discuss about the Craig plot and some of the references. So we are going to proceed in the same way. So first of all, we must know about what is the drug design, what is the concept actually involved in the drug designing method. Now, the principles involved in the drug designing method are: what is the purpose, what is the need behind anyone will go for the drug designing, is the improving the binding of drug. So we will improve the binding of the drug with the protein molecules, with the conjugate molecules. The binding with the protein molecule is totally different. Binding with the conjugate molecules, it is different. Binding of the drug with the protein molecules like albumin and all will increase the volume of distribution of the drug, and the binding with the conjugate molecules after its therapeutic effect will increase the excretion of the drug. That means, so the drug designing method, in a view of the same, it will improve the binding of the drug. Next purpose behind the drug designing concept is increasing the selectivity. So we need to go for target selection, and the drug must gives the selectivity pattern towards the receptor. The next is the drug must be will reduce the side effects, and whatever pattern for synthesis of drug we will frame out, the drug must be easily synthesizable in the laboratory scale. Then arrangement of the functional group and the identification of the pharmacophore. We will go for the arrangement of different functional group, which will give the better therapeutic index, and in a view of the same, we need to identify the actual pharmacophore of that molecular structure. So these are all 5 principles of the drug designing concept. Now, the drug design is actually based upon the lead molecule. What is the structure of the lead molecule? Whether the lead molecule is traditionally derived from any animal source, plant source, or from anything else that is actually obtained from the natural sources, or it is totally synthesized in the through artificial methods in the lab. What is the structure of the lead compound? What physico-chemical properties lead compound is going to have? Then analogs molecules, designing the new molecule, and some of the structures in which we will go for the different type of substitutions which will give the better pharmaceutically active ingredient or the pharmaceutically active drug. For example, salicylic acid is an analog, and from this analog we have obtained a drug molecule, that is aspirin. So this is an analogous designing which will give the aspirin from the analog structure, that is the salicylic acid. Now the aspirin is nothing but acetyl salicylic acid, which is obtained from the organic analog, that is salicylic acid. Now this is a drug designing method which is actually based upon the lead compound, on which lead compound the drug designing is carried out. Next is the drug designing, which also we will also go for based upon the target structure, on which target cell the drug is going to show its pharmacological effect, pharmacokinetic or pharmacodynamic effect. On the basis of that, we will go for the drug designing concept, and those by target structure is by identifying the structure of the drug target, by identifying the de novo drug designing. Then, and the third method through which we will go for the drug designing concept is basically based upon both leading compound and the drug target. What are those? The combination of both the methods, that is earlier, that is based upon the lead molecule and based upon the target structure. So lead molecule is belongs to the what drug on which the synthesis or the methods we will go for the application, and target structure is this lead compound is going to bind with which structure, which receptor, which macromolecule. That is nothing but the target structure. And the combination of earlier two methods, which is based upon either lead compound or on the target structure, this is a combination of earlier two methods that is based upon the leading compound and the drug target. Now, after discussing what is actually a drug design, on what basis the drug designing is carried out, whether it is based upon the lead molecule or whether it is based upon the target structure, we will go for one more quantitative method, that is quantitative structure activity relationship. Now this quantitative structure activity relationship approach attempt to identify and qualify the physico-chemical properties of the drug. By studying the physico-chemical properties of the drug, which will gives us whether any of these properties which will affect the drug biological activity by using mathematical equations. So by using some of the mathematical equations, we are correlating the physico-chemical properties with the drug action. So the correlation is based upon the mathematical equation. We are going to prove some of the mathematical equations that the physico-chemical properties is physico-chemical properties of the drug is going to have for its biological activity. Now, what are these actually physico-chemical properties which we are talking about since last 10 minutes? What are those actually properties which is going to affect the biological activity of that lead compound? And those are enlisted here. The physico-chemical properties are hydrophobicity of the molecule, hydrophobicity of the substituent, electronic properties of the substituent, and steric properties of the substituent. So 4 important physico-chemical properties which is going to affect the biological activity are hydrophobicity of the molecule and hydrophobicity of the substituent. Then what are the electronic properties possessed by that substituent and the steric properties given by that substituent. So these 4 methods or these 4 properties will directly affect the drug action. A range of compound is synthesized. If we are going for the drug designing compounds, in this particular methods, a range, that is diverse amount of the compounds, diverse range of compound is synthesized, and in order to vary one physico-chemical property and to test it affect the biological activity. So first of all, we have to synthesize a range or series of compounds. For example, we need to synthesize either same category, same analogs, minimum 20 compounds to go for the checking, to go for analyzing a physico-chemical property, whether it is going to affect the biological activity of that drug molecule or not. Now, these range of compounds, minimum 20 number, for example, minimum 20 numbers, then based upon the response of the bioactivity, a graph is drawn to plot the biological activity on the y-axis and the physico-chemical properties which we have taken, whether it is hydrophobicity of the substituent, whether it is hydrophobicity of the molecule, whether it is electronic properties of the substituent, or whether it is a steric property of the substituent. So based upon all these 4 properties, we are going to plot a graph of that is the biological activity on the y-axis and the physico-chemical property which we have chosen, it is on the x-axis. So it is necessary to draw the best possible line through the data points on the graph. Depending upon the results and the reading, we have to draw the best possible line through the data points which we have plotted on the graph. This done by procedure known as linear regression analysis by the least square method. So by selecting low number of the variables or least number of the variables, we are going to plot a graph of given data, that is biological activity on the y-axis and the physicochemical properties on the x-axis, which will give the linear regression analysis by the least square method. And after that, when we completed the drawing, if we draw a line through a set of data points, will scatter on either side of the line. If we will plot the line, so the results obtained by the lead molecule and the by the lead molecule, its biological activity on the y-axis and the physicochemical properties on the x-axis. So the data points will scatter on either side of the line. The baseline will be the one crossed to the data points. So the baseline must cross the 1 point from the data points. To measure how close the data points are, vertical lines are drawn from the each point. So we have to draw the vertical line from the each points in order to measure how close the data points are. Each our points are from each other. Now we will moving towards the first property of first physicochemical properties of the drug, which will affecting the biological activity. So the hydrophobic character of the drug. What is the definition of the hydrophobicity? So the hydrophobic character of the drug is crucial to use the hydrophobic character of the drug is crucial to how easily it crosses the cell membrane and may also be important in receptor interaction. So this particular phenomenon is actually useful to cross the cell membrane and may be important in the what are the receptor interaction with the drug molecule. Hydrophobicity of the drug is measured experimentally by hydrophobicity of the drug is measured experimentally by testing the drug relative distribution in the octanol water partition coefficient. So this particular method of measuring the hydrophobicity of the drug, that is concentration of the drug in the organic phase divided by the concentration of the drug in the aqueous phase, this ratio of hydrophobicities, that is partition coefficient, is first the physicochemical properties associated with the hydrophobicity, and this relative distribution we have given the name, that is a partition coefficient. What is the meaning of the word actually a partition coefficient? Partition coefficient of the drug is nothing but concentration of the drug in the octanol divided by the concentration of the drug in the water phase. Now activity of the drug is often related to the P, that is a P value. So what is the meaning of the word actually biological activity? Biological activity, that is 1 upon C is equal to K1 log P, that is log P belongs to log of partition coefficient value plus K2. So the binding of the drug to serum albumin by hydrophobicity and study of 42 compounds, straight line limited range of log P is obtained, and we have given the line which is actually passing best fit of the points. If the partition coefficient is the only factor, suppose we are going to compare the biological activity of any drug molecule with only one factor, that is a partition coefficient is the only factor influencing the biological activity. So the parabolic can curve we expect when we are talking about any one drug and the result shown by that drug, if it is going to hamper the biological activity, so the graph obtained must be a parabolic curve was expressed in the self equation. So log of 1 upon C is equal to K1 log P, that is partition coefficient 2 plus K2 P2 plus K2 log P plus K3. Few drugs where activity is related to the log P factor alone QSAR equations are only applicable to the compounds in the same structural class. If we will study about the QSAR methods of the same structural class, so the QSAR is useful. QSAR equation is very applicable for the compounds in the same structural class. However, log P0 is similar for anesthetic of different structural class. However, log P, that is partition coefficient of 0, that is initial phase, is similar for the anesthetic of different structural classes. So the substituent, that is hydrophobic concept. So the partition coefficient can be calculated by knowing the contribution that various substituent is going to have and is known as the substituent's hydrophobicity constant. In earlier slide, we have discussed about the concentration of the drug in the organic phase divided by the concentration of the drug in the aqueous phase, followed by we are moving towards what is the effect of hydrophobicity provided by the substituent on the biological activity. So the partition coefficient can be calculated by knowing the contribution that various substitution is known to subsequent the hydrophobicity constant, and a measure of substituent hydrophobicity, a measure of the substituent hydrophobicity is relative to the hydrogen. Partition coefficient is measured experimentally for a standard compound such as benzene with or without a substituent. So the hydrophobicity constant, that is pi x, is equal to pi x for substituent x. So the equation is pi is equal to, that is partition coefficient of the substituent is equal to log of px minus log of pH. Now a positive pi values, that is partition coefficient, substituent's positive pi value shows that the substituent is more hydrophobic than hydrogen. If the positive pi value is obtained after the equations, by solving the equations, will show that the substituent is more hydrophobic than the hydrogen. A negative value indicate that the substituent is less hydrophobic. The pi value is characteristic for the substituent, which will give the nature of the substituent. The example given here is benzene is going to have the log of p value is 2.13, chlorobenzene the log of p value is p.24, p2.84, and the third is benzamide is going to have the pKa value 0.69. So these are the various compounds and their examples which I have given, which we are using nowadays for the lab scale, and the pi values and the log p values are different. So the substituent hydrophobicity constant. Now the substituent hydrophobicity constant. So the log p theory, that is log of p benzene plus pi to pi x lambda plus pi x O N H, the value obtained is 1.57, that is metachlorobromamide alone. A QSAR equation may include both p and pi. So the measure of the importance of the molecule's overall hydrophobicity is relevant to the absorption and binding, etc. So pi identifies specific region of the molecule which might interact with hydrophobic, and the region is the binding site. So that identifies this identifies the specific region of the molecule which might interact with the hydrophobic regions in the binding site. Now, what are the electronic effects offered by the physicochemical? The electronic effects of various substituent will clearly have an effect on the drug ionization and property, have an effect on how easily a drug will pass through the cell membrane, and low slightly it can interact with the binding sites which is there in our cell. So Hammett equation constant, this is a measure of electron withdrawing, electron donating ability of the substituent on the aromatic ring. So depending upon the substituent, the person is going to have the Hammett equation is the measure of electron withdrawing or electron donating ability of the substituent on the aromatic ring. For example, for aromatic substituent, it is measured by for the aromatic substituent, it is measured by the comparing the dissociation constant of the substituted benzoic acid with the benzoic acid. So this is our norbenzyl acid, this is our benzoic acid, and which is measured by comparing dissociation constant of substituted benzoic acid. So the benzoic acid will undergo substitution, which will give the benzene, which will give the substituted benzoic acid from the benzoic acid, so which will give the benzoate plus H plus ion. So the KH dissociation is, so the KH of dissociation constant is log of phenol plus carbonate ring divided by phenol plus 0,4. So the electron donating group. So in earlier case we have discussed about the electron donating case. In this case we are going to discuss about the electron donating groups. So this is the structure of formic acid. This is the structure of benzoic acid, which will give the major yield of the formal compound, and the half say which will give the electron donating groups of the benzoic acid, which will give the benzoate ion plus H plus ion. Now this chain, the benzoic acid will undergo a dissociation constant which will give the benzoate plus H plus ion. So the charge detail equilibrium shift to the Ks is less than Kh. So charge dissociated equilibrium shift to the left. So the sigma x is equal to log Kx divided by Kh. So the log of sigma x is equal to log of Kx divided by log of Kh. So the benzoic acid will give the detailed distribution of the benzoate ion plus H plus ion. This particular equation will give us the now here are some of the examples in which different type of substitutions and what are the effects offered by them. So the first substitution is substitution of the amide functional group at meta position, that is withdrawing inductive effect. The next is para substitution. The drug and their substitutions are equally and symmetrically distributed. Those are known as para substitutions because they are substituted at para position to the available. Then para substitution and other examples are now the value depending upon the inductive effect and resonance effect. These values, that is electronic properties, are totally depending upon the electron donating groups, and the electron donating groups are electron withdrawing, that is inductive effect only. So here we have given an equation. Here we have given an acid, that is electron withdrawing inductive effect only. So if we are going to change anything at a para position, that is electron withdrawing inductive plus resonance effect. So values depending upon the inductive and values depending upon the inductive and resonance effect. Value depending upon whether the substituent is meta or para it is substituted. Orthometa values are invalid due to steric effect. Now what are actually this electronic effect? Electronic effect are R and F. R belongs to qualifies a substituent resonant effect, and F belong to qualifies substituent inductive effect. So one R will be substituted by inductive effect, and another will be substituted by inductive and resonance effect. Value depending upon whether the substituent is meta or para position. Ortho values are invalid due to steric factors. So the electronic effects are resonance effect and inductive effect. The constant sigma, R and F can only be used for aromatic substituent. So the aliphatic electronic substituents. Aliphatic electronic substituents, that is obtained experimentally by measuring the rate of hydrogen in aliphatic cases. Purely inductive effect given by PO, given by the sigma I, and hydrolysis rate measured under the basic and acidic medium. Steric factor. The bulk, size, shape of the drug will influence how easily it approach and metal with the binding site. A bulky substituent may act like a shield, and higher the ideal interaction between the drug and its binding site. Bulky substituents may help to orient a drug properly for maximum binding and increasing activity. Today we will stop here, and the remaining part of the QSR techniques we will proceed in the next class. Thank you.